Good afternoon. I'm really happy to welcome you to this special outreach lecture by Professor Ewan Birney. He's a co-director of the EBI, the European Bioinformatics Institute, which is part of the EMBL, to people that probably knows it. I know that many of you are familiar with the Ewan contribution to the field of genomics, proteomics, promoting new frontier in science. Let me share with you just one personal comment. I know that there are many young students here that for them, you know, deep sequencing is like the routine. It's like everything sounds as if it was forever. So it wasn't like that. I think I met so all this concept of big data and informatics leaders is something that is happening while we are talking. And just to say, I'm Michal Imyal, I'm the director of this institute, the Israel Institute for Advanced Studies, and also the representative of ELIXIR Israel. I hope you'll hear a little bit more about ELIXIR. I think many of you that have been in the bioinformatics annual meeting already heard about ELIXIR, and this is really I won't talk much about ELIXIR, but I'll say that this is the pan-European consortium or initiative really to build up what's needed to be done in the next generation of science and the health science and so on. I met Ewan about 10 years ago already with Janet Thornton being the head of the EBI, and really on shaping the idea what and where are we heading basically, and I think the talk will be exactly on this phase. So just a few words about Ewan. He completed his PhD at the Wellcome Trust at the Sanger Center, Sanger Institute with Richard Durbin. I know that many of you that study bioinformatics know that Durbin is the kind of the Bible, so this is the Bible that everybody studies from this very important book. And Ewan received the Francis Crick Award from the Royal Society. He received the Hoverton Prize from the International Society for Computational Biology, ISCB, and really Jonathan Franklin Award, and for promising for open source, which I think is very important also aspect of what we are doing. I mean, really to share the community with what we are doing. And Ewan is recently elected as a fellow of the Royal Society of the Academy of the Medical Science. And for the young students, I'll just say that projects such as ENCODE and 1000 Genome and Ensemble are all part of what Ewan really promoting and pushing for the last decade. So, toda rabba and welcome.
Okay, thank you very much. It's really lovely to be back in Israel. I was here 10, 15 years ago, younger then. I feel a bit like an old man. And you haven't changed. It's sunny. Jerusalem is still amazing. Israelis still argue. So there are these constants in life. So I apologize in advance if this talk can either overshoot or undershoot, depending on your own level here. So I apologize if you find this too simple or too hard, and I hope there's a little bit for everything. So I'm going to talk about some strategic stuff at the start. I'll talk about ELIXIR, some infrastructure and medicine in the middle, and then I'm going to end up with something fun at the end. So if you're getting very bored, just hold on until the end. So we've been living through, it's been a really remarkable decade if you're a computational biologist or if you're a biologist of any sort, actually. This is my way of describing this change. Back in 2003, an American individual, Craig Venter, persuaded the American stock market to give him enough money to sequence a human genome. That ended up being his own genome, by the way. And that was about £26 million at the time, which was the same amount of money as the most expensive house in London. And this is a picture not of that house. I don't think you're allowed to take pictures of that house, but it's a house on the same road as that one. These days, if I wanted to get my genome sequenced, actually an Arsenal season ticket would cost me more than getting my own genome sequence right now. There's a lot of people with an Arsenal football season ticket. Next year, it will probably be a West Ham season ticket. I have no idea what the price of the Tel Aviv season ticket is, but there are, all of us will actually spend more on going out to a restaurant, perhaps with our family, over the next couple of years than it would be to sequence your own genome, at least for an exome. So that's a remarkable change in cost. Really, really remarkable change in cost. Now, the driver for this technology change has been human health. The reason why people have made sequencing become so cheap is because of human health. I'm going to spend quite some time on it. But if you're a molecular biologist or a biologist, you'll know that genomes, DNA, RNA, and proteins, the stuff of life, is very, very ubiquitous. It's very much the same. So despite the incredible diversity of life, the trees outside, the fish, everything else, they look completely different. They are made up of the same stuff. So the technologies developed to sequence and understand humans, you can apply to agriculture, and you can apply to everything that's alive. If you're an ecologist, do we have any ecologists in the room? If you're an ecologist, all your Christmases have come at once. You've got a completely new way of surveying ecological systems by using these measurements. And the other big shift is that research itself has changed. So I used to do this, molecular biology lab work. It's basically advanced cookery. You have to be very careful, very precise to achieve some ends. This still is important, but it's now equally important with this computational work as well. So an awful lot of research now happens by thinking about the data. There's dumb things like keeping your data straight and cleaning it and stuff like that, stuff that not a lot of people talk about because it's quite boring. And then much, much more exciting things, which is how you actually conjure up knowledge out of large data sets. I'll come on to our impact on medicine and how this is changing, but I just want to stress that it's way more than medicine. It's quite possible that genomics will have a much bigger impact through agriculture than it does through medicine. Quite a lot of crops used in the world haven't been through any proper optimization and yield management, yield selection, and genomics really, really narrows or shortens that time. And then this is the latest whizzy ecological thing. These are a bunch of mad Frenchmen, mad French ecologists that sail around the world sampling the world's oceans and then sequence the results of that very, very deeply. And that told us all sorts of things about the oceans that we never knew about before. So there's going to be a bit of a theme. As I mentioned, this is a picture of a very important but unloved technology, which is London sewers, Victorian sewers. If you live in London, you're kind of dependent on these things, and of course you don't want to talk about it a great deal. And there is a similar sort of aspect, not quite as smelly, in bioinformatics, which is called kind of data plumbing and keeping all your data straight. Many, many, many projects fail because they haven't kept their data straight over the lifetime of the project. Of course, people don't put their hand up and say, oh, my project was a failure because we forgot to structure our things correctly halfway through. We just lost track of things. They come up with much more exotic reasons for why their projects fail. When you get underneath it, you discover that they just didn't manage their data particularly well. What's much more exciting, I'll touch on this, and I'm also giving a talk tomorrow at the Weizmann, so if you're Tel Aviv way, come there where I'll talk more about work that one of my students have done about big data, high dimensional statistical analysis. And though I illustrated that with a sewer, I actually think it's much closer to the engineering like this. This is CERN, the Large Hadron Collider in CERN. And in fact, bioinformatics and big data in biology have become close to the levels of high energy physics. We're just one order of magnitude smaller than high energy physics. Now when I go and visit CERN, and it's kind of fun, it's very geeky, you hang out with all these people, I cannot understand the physics they're actually trying to test. I mean, I can say the word Higgs boson, but I really don't understand it in a deep way. You know, there's a nice little graph with a bump, but that's about as far as I go. But the engineering, I find the engineering fascinating because I can understand the engineering. So they have this 26 kilometer ring. It's a superconducting magnet, and they obviously that has to be kept cold enough that it remains superconducting, and they use liquid helium. And I said, oh, that's very interesting. I thought superconducting magnets worked at liquid nitrogen levels. Why do you use liquid helium? And so the engineers explained it, and there's two things about this. So the first interesting thing is once you get to the insulation level that you can store a lot of liquid nitrogen, it's actually not so hard to drop the temperature down to liquid helium. So in other words, it's all about your insulation. It's not really about the Kelvin aspect. But the main reason they use liquid helium is that the superconducting magnets here have an awesome amount of energy going through it. And if it's 26 kilometers in diameter, and if a single magnet, single part of that went non-superconducting, the thing would explode completely, I mean dramatically. So now you've got this engineering problem. How do I keep 26 kilometers of stuff below a certain temperature and really guarantee that you do that? And that's why they use liquid helium, because liquid helium is a Bose-Einstein condensate, and that means it has one quantum state across all 26 kilometers. So they've used quite a fancy piece of statistics, the fact that liquid helium has this property, so that they can just actually measure the temperature in just one place, and by quantum physics they know that that temperature is the same temperature across the entire ring. So I actually think that bioinformatics problems, the plumbing is getting interesting. So there are aspects of the plumbing which you think aren't interesting, and then you have to face these big challenges and they become interesting challenges. So just to mention a little bit where I come from, which is EMBL-EBI. As Mikhail mentioned, we're part of the European Molecular Biology Laboratory. That's an international non-profit research institute. Israel is a signatory to this treaty. Israel helps funds us. You're one of our 22 nation states. So in some sense, your taxpayers are my boss. Or you guys are my boss via tax there. We're Europe's hub for biological data services and researchers. We're quite big. We're actually, this slide is out of date, with 600 people. There was a big argument back in 1992 how big the first building should be, and whether we'd ever grow above 80 people. And clearly we have. There's more than one building now. And I think that gives you another sense about trying to put some of these things in perspective. So what do we do? So one aspect is deliver services, and this is probably what you most think of when you think about the EBI. So life is full of all sorts of different molecules. And it's not really that actually physics is full of all sorts of different things as well. So some people make a big distinction between physics and biology here. I tend not to. But there's no doubt that the heterogeneity in biology is much, much higher. In other words, we like to study all sorts of different things, and we study them using many, many different types of measurement. And all of these different things we can measure. And the other interesting thing in biology is that you spend an awful lot of time, we use what you might call ground truth data a lot. So I'll take an example of the human genome. The human genome, we have theories about how it arose via evolution. We have theories about how it creates information that's used in the cell, things like making RNA and stuff like that. But our theories don't tell us what the human genome is. You just have to go out and determine and measure the human genome, and then you have to store that information and pick it up again and again and again. So that process of storing ground truth facts and giving them back out to people over time is the core part of our service delivery. And those map to different sorts of databases. Mikhail mentioned Ensemble, which I helped found. There's all sorts of other things, ENA, which is the DNA data. This is the metabolomics, Uniprot. Chembl, which is small molecule drugs, associations of small molecules that affect proteins, KEBI. We've got pathways, literature, all sorts of different things. And these things really form an infrastructure And infrastructures are quite fun. So when I came here yesterday, I used this infrastructure and this infrastructure. This morning I used this infrastructure, and I'm using this infrastructure now. And of course, what I didn't do, I didn't phone up Mikhail and say, okay, on Sunday, is there going to be electricity in Jerusalem? Because if there's going to be no electricity in Jerusalem, my talk's not going to work, I'm not sure it's going to be a very successful day, I think I'll cancel. I made the assumption, correctly, that the infrastructure of Israel was going to continue to power, correctly, I noticed the smiles, that the infrastructure was going to correctly power this system, and I mentally forgot about it. I mentally forgot about it. And in fact, we only really notice infrastructures when they fail. So good infrastructures disappear. Good infrastructures, you mentally skip over them because you don't worry about them. So this is obviously cataclysmic failure. This is Heathrow in the snow. I'm sure you're all familiar probably with this kind of infrastructure failure. A very small dusting of snow in Heathrow creates a kind of 2-week scheduled disaster zone after it. Whereas if you go to my colleagues in Finland and Helsinki, they will keep on trucking all the way through the winter. There's a difference in infrastructure provision with respect to snowfall between Heathrow and Helsinki Airport. This is a bridge in America, and it's a town. One half of the town is over here, another half of the town is over here. One bridge, the next bridge is about 20 miles in one direction, the next one is 30 miles in the other direction. The collapse of this bridge caused a real problem for the people living in this town. Again, a piece of infrastructure failure. So our science demands now an information infrastructure. You've probably written, if you're a PI, you've probably written grants where you've said, I'm going to compare my data against the human genome. And you haven't said to yourself, just as I didn't worry about electricity, you didn't say to yourself, ooh, I wonder if the human genome is going to be around for me to compare against in 3 years' time. You made the assumption it was going to be there. Many of you might have used Go terms and Go enrichments of these things, or you might have used 1,000 genomes or exact to clean up your clinical medical data for these things. Again, when you've written this down, you haven't worried about whether these things exist or don't exist. You assume it's going to be there. And this continues to happen in the future. And when you think about it, this gets quite scary. Because biology is endlessly diverse. Everything which is alive that we might want to study has the same properties that we'll want, these reference data sets that we'll want to use and understand. And we are not, at the end, it's not like the human system is somehow solved by having the genome. There are many things, there's huge amounts of unknowns where we don't understand those facts. And so we've got in front of us 50 years, 100 years of carefully determining facts, understanding them, storing them, giving them back out to people. I should have put this picture in earlier. This is a satellite picture, a real satellite picture of an infrastructure failure. Isn't that a beautiful, a remarkably shaped system on the US system where they had a kind of cataclysmic grid failure that actually triggered a new investment into smart grids by the US to prevent this ever happening again. Actually, it's quite a scary couple of days for that part of the United States. So one of the things that became clear to Janet and myself and my colleague Rolf Apweiler 7 years ago was that this infrastructure that we feel that we provide at the EBI, that we weren't going to scale well. And actually, this isn't a computational problem. I'll come on to some of the computational aspects of scaling and storage and stuff like that. And that's mainly an engineering task, and you can either do it or you can't do it for a certain amount of money. The thing that we really weren't going to scale well was expertise. And so that's going to set up, I'll have a little story about this in a moment, that led towards the business of ELIXIR, which is, from our perspective, broadening the infrastructure provision out just from the EBI across all of Europe. And that has other benefits as well as actually providing it. it means because ELIXIR is a sort of multinational project, it was launched in Brussels in 2013. Israel is on this lovely list. 17 nations, is a little bit out of date. Not only did 7 nations sign the MOU, but a number of them have committed to make the entire thing work as well, including Israel. So we now have a kind of operating system. And I'll just do this example here from Bergen. So cod, ocean-going fish are quite important. We fish them, we eat them. We don't really know much about them. Would you believe we don't know how to keep a cod? We can't grow a baby cod to an adult cod in the lab. There's certain parts of its life cycle which we don't understand what they actually eat. We can't provide it to them in the lab. So there's all sorts of research you can imagine doing on cod around fisheries. Now, at the EBI, we're very close to Cambridge, UK, Cambridge in the UK. And if I went to Cambridge, I would not be able to find even someone in the top 100 ocean-going fish genomicists. I know, I've tried. So nobody in Cambridge is that in to ocean-going fish. If you go to this institute in lovely Bergen, you find 4 or 5 of the top 10 ocean-going genomicists, ocean-going fish genomicists, are all situated in this single institute. So for the Norwegian ELIXIR node, they have chosen fish genomics as their kind of aspect that they are contributing to the international aspect of ELIXIR. They're also going to do a number of things around human health, and most ELIXIR nodes have this balance of 2 things, a kind of health focus and a non-health focus, and there's this sort of hilarious business that the national icons in each country often get picked up. So the birch tree, the Finns obsess about the birch tree for all sorts of things, lots of birch tree forests, also sauna, big part of sauna. And so they really wanted to do the birch tree genomics. And we did say to them, that's a little narrow. Could you think about it a little bit broader? So they've gone for tree genomics, which is a bit better. And obviously there wasn't a lot of competition. Like nobody else put their hand up and said, I really, really, really want to do birch tree genomics. Same thing about the truffle, all sorts of different things. In human health, this isn't so sort of, you know, every country wants their citizens to be healthy. Every country wants to understand human health better. Every country wants to deliver this for the people inside of them. But I'll come on in a moment that, again, the health drivers to this produce another different national perspective. So just to mention, at the EBI, we not only do service delivery, we also do research. I'm not going to spend an awful lot of time on this, except please do come and read some of our research. And if you have time tomorrow, come to my talk at Weizmann, because I will give you an example, really, of some research into human hearts, healthy human hearts. So I'm now going to come on to thinking about medicine, and I now ask a question: How many people connected to medicine are in the room? I know, in case it's a fair number, not so many. Great. How many are actually qualified clinicians? Okay, right, that's good. More than 0. That's my key thing. So the first thing to understand about medicine is that it's very, very different from research. Medicine, research is practiced in a very different way from how healthcare is delivered. So we use English as a language. Israel is in one of these countries where you can do it both ways. But for most countries, healthcare is absolutely delivered in German. You do not expect in Germany to have your doctor try and talk to you in English. You expect your doctor to talk to you in German, unsurprisingly. It's got very lightweight legal systems. The default is for open data and publications, and most of the money are provided as grants. And by what I mean by grants is that the person on the other side who takes the money, as researchers, we promise to do our best effort to say what we do. But if we don't deliver the Nobel Prize or whatever we've promised in our grant application, we don't get sued by the funding agency. There's not punitive damages at the end of the day. Practicing medicine is very, very different. In fact, it's one of the most nationalistic parts of our economy. The European Union has an article that specifically prevents the European Commission from meddling in healthcare systems across Europe, for example. So it's one of the most national systems. It is very, very different as you go from nation state to nation state. How medicine, disease biology, type 2 diabetes is the same here as in England, as in France, as in Denmark. What actually happens to you when you get type 2 diabetes is actually very, very different in those different systems. And just to mention, the data is closed, it's not published, and there's contract funding. In other words, there are punitive parts of the terms if you don't come up with it. And the systems are really wired completely differently. This is the one I know best, which is the UK NHS system. It's a single payer system and it's a single organisation, and there's this standardisation. I will not attempt to read this very long German word here, and if I could even get my computer to type the Hebrew word for healthcare HMOs, I would have done that. So it's just completely wired differently. It's a very separate thing. So it's really clear to us at the EBI, it's clear to us that our science, but also our data, is becoming more and more important for medicine. And I'll give you the classic, iconic example, which is the human genome, the reference human genome. We are part of the international consortium that looks after it. It will improve. There are errors in it. They do get fixed. There will be progressively better things. And we need a way, a communication channel, to sort of flag to the broader set of healthcare systems around the world, the human genome is going to change in 2 years. These are the different reasons why. Here are the regions to really worry about. Here's the stats to show you that it's better. All of these things. We need a way of communicating with this. But we have to end. It's very clear that we are not the full solution. There has to be something else that takes that. And it's also extremely clear that that cannot be, for example, individual research intensive hospitals. There have to be national systems that sort of think about this information. So ideally, there should be, in our view, an Institute for Biomedical Informatics in each EMBL nation state that we interact with. This is our ideal. Now, in bigger nations, this is very likely to be a network with a center of gravity. In Israel, you can all probably drive for one meeting to one place, just about, maybe. I think with smaller nations, you have there's different challenges, but also different solution spaces are open. EBI and ELIXIR, we will handle the research-style data and to be shared between researchers, including clinical researchers. So that's the role that we see the EBI and ELIXIR infrastructure. And I should have added here, in smaller nations, I think it's one of the opportunities that smaller nations have, Denmark, Estonia, ELIXIR, Israel, and other places, is to think about putting biomedical informatics and ELIXIR very close together. There aren't so many places anyway in Israel you're going to put these things. So more coordination rather than less is probably a good thing. This is definitely an Israel decision. And then what we'd like is a two-way piece of communication. And so that's both about exploiting molecular reference data and basic understanding inside of countries, but also the ability to broker research-style data back to ELIXIR. And I'll just mention this, which is that there's a big grey area kind of in the middle of the bridge. And this is about humans becoming a first-class model organism and us studying humans to understand humans better, but frankly to understand life better. And so in many places there are cohorts of patients that have been put together from different things, but this cohort both provides a research face and provides a healthcare face. And so you have a dual-facing system where the same group of people are being used for research, and that is part of healthcare delivery. So probably the country that is furthest along with this and most most visionary in many ways is Iceland. Now, in many ways, Iceland is 300,000 people. There's one major hospital. It is not the hardest nut to crack. But Carrie Stephenson realized this 10 years ago and put together with the great genealogical databases in Iceland. They really kept their parents straight for a very long time into an amazing resource. Denmark, Faroe Islands, Finland, the UK Biobank, I should have put the UK 100,000 genomes, and Estonia. These are all really interesting countries that are exploring this at the moment. I think the French government will be doing this soon, not least because they want to, as ever, be better than the British, which is a very, very good motivation for the French. And so the British have the 100,000 genomes. I think they might have the 100,001 genome project in France. It's all recorded. Yeah, gosh, I'm sure the French will. Ooh, that's interesting. The French, I think, will agree that they're better than the British. So that's easy. So as I mentioned, humans are we are good model organisms. We are really worth studying. So some of the features about humans as a research organism. So we're outbred organisms with pretty good genetics. In fact, seemingly, the only real issue about our breeding patterns is geography. So don't tell that to my wife. But we seem to mate randomly when we meet, basically, from a genetic perspective. We're big organisms, and you can get substantial amount of cells from us. Now, that doesn't sound so serious until you do some bits of molecular biology, in particular blood cells. So obviously the human white blood system has been studied extensively because of this. But you can actually get quite a lot of cells if you've got some pretty brave volunteers. You can get some substantial amounts of adipose and muscle and all sorts of other things. We walk into these already funded phenotyping centers. These things are called hospitals. And if you tickle the hospital right, you can sometimes get good phenotypes out of the hospital. And that process of tickling information out of hospitals is a bit of an art at the moment, and that needs to improve. And we also attract a lot of funding. So quite rightly, quite a lot of the funding agencies are semi-obsessed about making us healthier, and therefore it's easier in many ways to make the connection. In fact, I don't think it's slightly overplayed. I think lots of diseases will be solved by just studying yeast, understanding a molecular process in yeast, and then walking straight into human and taking that understanding and solving some things in human. So this shouldn't be overemphasized that the model organism for human disease, but there's no doubt that the connection between studying humans and human disease is much, much closer than with other organisms. And this is one example. This is an example that my students, sorry, the third time I'm going to mention this, I'll present this properly tomorrow, where we're studying healthy human hearts. This is the left ventricle of human hearts, and this is an association study where we now get some points. If you haven't seen these plots, if you're a statistical geneticist, you look at these all the time. It's called a QQ plot. And this basically says my genetic test was sound. That's because the black line is on the red line at the start. And then there's something interesting up here, and that's because the black points are above the red line significantly. And interestingly enough, in this case, we were able to untangle what this particular genetic variant was doing on the human heart. And it ends up being that we have found different morphotypes of human hearts, of left ventricles, that are present in the natural population, and we're exploring more what this does. This is actually a chromatin modifying gene. It's very clear that it's involved in some developmental program behind the setup of this. You could have done this in mouse. There's a mouse experiment that actually was done, but it's not so easy actually to get 1,000 mice, outbred mice, and do something with it. It's actually arguably easier to get 1,000 outbred humans and put them into a study than 1,000 outbred mice. And so there's all sorts of things we can do in humans which are actually harder to do elsewhere. So just to mention our role here, this is EMBL-EBI's role for this, which is proactively transferring our knowledge and skills into the healthcare sector. So one slightly tedious plumbing thing, which I'm sure you're going to face, is how to store lots and lots of DNA, perhaps all of Israel's genomes within 5 years. And we have technical solutions which compress the data far better. It's a mundane point. This is not particularly sophisticated, but you might as well profit from what we have done for this. But the more interesting thing is forming a return loop of both information and knowledge from the basic side and our understanding of life towards clinical research and practicing medicine, but also for practicing medicine and clinical research to be returning knowledge that can then be used across the entire world around disease biology. So, I promised you something fun at the end, and so if you found that all a bit dry, this is much more enjoyable. So I'll just set this up as one of these engineering problems. This was in fact what I just mentioned, this compression. This is, by the way, just to explain the plot, the x-axis is time, and the y-axis is log of the number of bytes. So we don't put these things on a linear scale. This is the amount of storage the EBI uses. If we put it on a linear scale, you just get a very silly graph that just kicks right at the end and goes up too fast. So a straight line on this graph means an exponentially increasing amount of data. And what we worry about actually is not really where you are on the y-axis, but in fact the slope. Because if the slope is 18 months or slower, we don't worry about it. And the reason why is disks improve in capacity at about every 18 months. Every 18 months, they double their capacity for constant money. When it's 12 months, we start to worry because we are going to be consuming an exponentially growing amount of money in our disk to do this, disk budget, and eventually we'll have no money to do anything else except store this unless we solve this. Now, looking at that plot led a number of us to form this compression method, and we actually can get up to 100-fold better compression if you accept some level of lossy behavior, and that's been rolled out into production now, and it's the solution. That was our engineering solution. We're quite happy. But 6 or 7 years ago, look, if you think it's scary now, have a look at that point. At this point, my colleagues in EMBL were sort of yelling at me that I had to give it up. We weren't going to be able to store the world's DNA. Just stop it, Ewan, and we're going to use the money for something else. And at the time, we were having a joke, myself and Nick Goldman, over a beer, that that biology had made this beautiful digital storage molecule, and that beautiful digital storage molecule was, of course, DNA itself. And so over a beer, so let me just remind you about DNA. DNA is a chemical. You find it's two polymers that hydrogen bond together, and those polymers are made out of 4 options of monomers. So there's 4 monomers that you build this polymer out of, and these are the chemical structures, and we've discovered that Lego bricks are the easiest way to explain this to people who aren't used to chemistry. And so in DNA, you're allowed to have a standard set of the bricks fit into each other, and now you can build any kind of polymer that you like with these 4 different colored bricks. And it's kind of obvious in inverted commas that this could store digital information, not just information in our cells about how our cells work, but anything that we would like to put in, we could store in this. And the key thing is that you can actually make DNA to your own design now. So this is nothing. It's a company called Agilent, and it's basically like a giant inkjet printer. But rather than printing ink, it prints these chemical monomers, and it builds it up on a little spot, and eventually you get a piece of DNA of your own design. So over our beer, Nick and I talked about, how feasible would this be? So how feasible could we store not just DNA information in DNA, but if we could store information in DNA, why don't we just store anything, any digital information in DNA? We realized pretty early on that this was doable, but the major problem was going to be error correction. So when we sequence bits of DNA, we tolerate a certain level of errors, and we sort them out in a variety of ways. When we make DNA, we tolerate a certain level of errors. But when you store a digital file, you probably tolerate 0 errors. So if you're a computer scientist, you'll put your hand up and say, you know what, you can never promise 0. So let's give, a very, 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 very small number, which is above 0 for this. And that's what all your mobile phones do and your disk drives and everything else. And when they fail, it's usually because one part of their error correction routine, there's been enough errors that has pushed past some boundary in the system. So we were thinking about the error properties of DNA, and we came up with a way of, so this is called a codec, where you have digital information and you convert it very often into another piece of digital information that you push over a channel or something like that. That would be in your mobile phone. In this case, our channel is DNA fragments. And we decided that our coding system would have so we realized that homopolymers, the same letter, one after another, would cause both encoding and decoding problems. So we created a codec that has no homopolymers, and as it happens, that goes through a base 3 intermediate. Now this was and then the other aspect of this is that you cannot synthesize very long pieces of DNA. You can't do that in one of these inkjet printers. So what you can do is synthesize short pieces of DNA. Now if the molecular biologists in the room might say, well, this is fine. I could take the short pieces and then synthesize them with an overlap, and I'll do a bit of PCR, and then I'll get another long piece of DNA. But if you think about that, then you have to keep a long piece of DNA. That's very difficult when you do liquid handling. They all shear and shatter. And for the computer scientists in the room, you might say to yourself, sod this, why are we trying to make a long piece of DNA at all? Why not just make short pieces of DNA, but have pieces of information that tells me where they are in the bigger message? So we make these short pieces of DNA with a little header, it's like TCP/IP packets, to tell us where the DNA is. So at this point we were on our second pint of beer, and we went home and we thought about it, and Nick said, yes, I think this will all work. And so I said, let's go and do it. And I rang up Agilent, which is this company in California, and I said, you're crazy Californians. Could you please do this experiment for us? P.S. We don't have any money for this experiment, so could you do it for free? And thankfully they said, we're crazy Californians, this is just the sort of experiment that we like to do. So we had to decide what to store. So we stored this picture of the world's leading bioinformatics institute, just south of Cambridge. We stored this MP3.
That one day this nation will rise up and live out the true meaning of its creed. We hold these truths to be self-evident, that all men are created equal.
It's kind of great. Something that I didn't realize until later on was that he had a very different speech prepared. He had written down a very different speech. It was a less sort of visionary speech, but then he saw the number of people who were coming that day, and he put the speech to one side, and he ad-libbed. He probably had done it a number of times before. So he had not written those things, those words beforehand. They were really in front of him, just came at the day. We stored this PDF, which has the famous paragraph. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So one of the famous PDFs of in DNA. And we stored all of Shakespeare's sonnets. And I wanted to show all of Shakespeare's work, but then Agilent said, you're going to have to find some money if you went to that. So we decided to just go for all of Shakespeare's sonnets. And DNA is a really remarkable molecule. It lasts longer than all known human written work. We have successfully recovered DNA from a dead horse in Alaska. The horse didn't say, ooh, I better die in a very clever way to store my DNA for 700,000 years. It just died in Alaska. If you think about it, we are going to be achieved. That's before modern humans evolved, by the way. So that's the kind of lifespan of that you can store DNA for. It's incredibly dense. You could actually get the entirety of the internet, that's a couple of zettabytes, into a small truck. Now that would be impractical for a couple of reasons. So firstly, if you wanted to retrieve any information, you would have to sequence 3 zettabytes of DNA to find anything, and that wouldn't work. So clearly you wouldn't just store it as 3 tons of DNA in the back of a truck. You would have some kind of indexing system to do this in practice. But also at the moment, all the money in the world would be going to Agilent to achieve this if we wanted to synthesize that amount of DNA. And so clearly that's the thing that's got to change. The other really interesting thing, this is very different from all other digital materials, is that although making the first copy of DNA is very expensive, making future copies of DNA is cheap, really cheap, stupidly cheap. In fact, in many ways, it's as simple as just pipetting, because most times when we talk about DNA, we don't talk about single molecules. We talk about solutions where you have millions of identical molecules in the same thing, so you can just split by pipetting. But you also have PCR or isothermal replication. And that's a really interesting property of this digital material, which is that you can make copies at will of it. So thank you for listening. I talked about serious stuff at the start, and then I was much more frivolous at the end. I'm extremely happy to take questions. You can follow me on Twitter, and I blog regularly, so do keep in touch. And I'm here to help stimulate ideas about Elixir, but also about bringing Elixir and biomedicine closer together as they are happening, I think, all across Europe. So thank you very much.
Thank you very much. So Ewan agreed to answer as much as we want, basically. So please, and in order for it will be recorded, I'll pass the microphone.
When you showed the picture of the sewers of London, I thought you were going to say you're going to sample the sewer and sequence it. And then I thought, wow, that's actually not a bad idea.
That reminds me, I should show you, I have another prop. Yes, people are doing this at the moment. So there's a sewage works that are sampling their sewage, especially in the bioreactors. So there's a very interesting thing that the bacteria suddenly don't work. People don't understand why. So sequencing sewage works is a very valid thing. And just to say, technology is continuing to improve. So I am a consultant for this company. But this is one of the latest sequencing machines, which you can plug into a USB device. And so you can think about sampling rivers or sewers or sewage plants or your food chain or your hospital or your blood every day.
How about your own toilet?
Yes, that's being suggested. Yeah. So you have to start thinking about DNA not being expensive, complicated, the hardest thing in the world to do, but being trivial, cheap. Very easy. You can hand that, as long as this comes back to me, okay? Right, it doesn't actually work, I'd stress, but I like it as a prop. So do fiddle around with it. This is actually the sequencing bit of it here. This is basically a thermal heat sink, stuff like that. You can take it apart like this. And it has very nice magnets. Okay, so have a little play. So that's a DNA sequencer that would sequence a bacterial infection in a hospital in under a day.
Questions? Any questions? So maybe I can ask till you warm up. If you have to guess, 5 years from now, just guess. Again, no penalty, as you mentioned, so you can do whatever. What type of information will take the biggest chunk? Is it visualization, genomics? I mean, what we expected, as a guess?
So I do think, and I think this is true in Silicon Valley as well, which is that data management at scale is becoming harder and harder and more complex. And it's very unglamorous. It's an incredibly unglamorous world. And by that, I don't just mean storing large amounts of data, but being able to do it in a way where it's very heterogeneous. You can get the data back that you want, with different. So this is about ontology management and all these sorts of things. So I think that's not going to be the biggest amount, but I think there's going to be a massive amount of challenge around data management. And then the other challenge is around understanding it, and this is just high-dimensional statistics. Yeah? Except, as if you're a statistician, you'll know this, there is not like some kind of magic, this is the way you do high-dimensional statistics. Yeah? You have a sort of a set of different things you could do, you have a set of different problems, you need to think about how you cast your data into something around this. And I really believe that there'll be new ways of doing high-dimensional statistics inspired by these problems. So I do think that computer science and statistics needs to come, we need to go back full circle. If you know frequentist statistics and Ronald R.A. Fisher, a lot of statistics was inspired by biological problems. We need to go back to that world where biology is as much a driver for statistics as is high-energy physics or financial or epidemiology.
Yes, please.
So you nicely showed the variety of data that we handle and the size of it, and you spoke about engineering solutions and other things, but not in the integration between variety of data. Yeah. What's your vision of integration between different types of data?
Yeah. So it's a good question, and I don't think there's a single answer to them. So I'm actually going to repeat what I've just said again, but I think it actually has an extra twist for the kind of what you might call multimodal integration. So one is a lot of projects fail because they don't have their data straight. So the sample measured by A wasn't connected with the same sample measured by mechanism B, and so they fail right at the start just by this connectivity process. And you'll be surprised how many projects fail that way. Again, it's not something people like to talk about it because it sounds so stupid that people don't put their hand up and say, well, I wasn't able to do this because we just didn't keep our data flow straight right at the start of the project. But then, assuming you've done that, which most people would say, when you write your grant proposal, you won't say, I'll have really messy, complicated data, and my postdoc will spend 6 months trying to sort it out. You won't write that. You'll say, my data's all going to be fine, and we'll separate them out into the right things that will be connected. Then you have this extra problem about how you combine multiple high-dimensional data sets together. And it's as if there's another line to this. So, unfortunately, now there's no generic answer. It's not like, oh, you bring out Bayesian statistics and form these kind of models, or you'll do it with deep neural networks. You just have to try, you have to work out which part of your toolkit fits those problems best. And it's extra challenging when you go between different data sets. Final word of warning, and another sort of aspect of this is, and again, every bioinformatician, computational biologist will know this, all high throughput measurements have systematically, wonkily measured things. So what you might call artifacts, they're actually using the word artifact is also a bit misleading because they're perfectly reproducible. They're parts of the measurement process. They're just not what you wrote down in your model about what's going on. Yeah? And so understanding those measurement specific, let's call them artifacts for the moment, is really, really important. And a massive, too much of computational biology is spent discovering those things and excluding them so that your central part you can compare validly, as it were. And I don't think we do enough, we're not good enough in sharing those processes. So again, they're not talked about a great deal in papers, and then everybody has to go off and recreate the way you clean up a ChIP-seq experiment correctly. And it's a bit frustrating, really. So I think we need to be a bit better in our community in sharing our data cleaning techniques.
Is ELIXIR going to take part of exactly this?
I think what I think is great about ELIXIR is it comes from the perspective of supporting and enabling science. And this sort of data cleaning thing is exactly the sort of unglamorous thing where people say, oh, I assume somebody's done this well, could I roll this out? I think virtualization is very useful here, so you can imagine about little Docker components and stuff like that. But none of that escapes the fact that you need to have people. So I think the other key thing about ELIXIR is having trained individuals who have kind of seen these kind of data sets before coming through the system. We need to have good human capacity in this area.
Please.
So you stressed how important it is for these bioinformaticians and those who are working with them to know this. Is there an educational component to ELIXIR?
Yeah, there is. There's a training component indeed. And I think when we talk about ELIXIR and training, we very often talk about training. There's sort of two things that the word training is associated with, and both of them are very, very relevant. So one of them is training about using our databases and tools, and that's kind of slightly, it's not, that's very targeted and focused training around these sorts of things. But then there's this other thing, which is having people who think both biologically and computationally. So that's more about master's students and PhDs and stuff like that. And to, in fact, encourage a whole career path for people who think computationally as much as they think experimentally. So, some people use the word training to mean all of this. Some people use the word training only to mean start. Some ELIXIR nodes only focus on this bit. Some ELIXIR nodes focus on the whole thing. Both are needed is the key thing. So this isn't just about training to use tools and databases and methods, so that's incredibly valuable. It's also about producing the right interdisciplinary skills, you would call them, for future generations.
Maybe I can just add on ELIXIR Israel is committed to education and training, mainly because in almost all university, already in the last 20 years, we have programs more successful, less successful, but we take it as a undergrad training, and I think in this audience there are quite a lot of people from this training system. So it's definitely a very big component of what we are committed to. Yes, please. Ilan.
One additional related question. So you showed nicely Turok of DNA as a metaphor, but one thing that academia is oriented to discovery and not to infrastructure. And as Phil Bourne likes to show, within 2 years, 2/3 of all software servers are lost. Any repository and system administration for that, because right now, if you want to submit a structure, you have to save the coordinates in the PDB, but if you want to submit a paper with a new URL, nobody keeps track whether it will keep alive.
Yeah. So I think understanding the balance between long-lived infrastructure and more experimental things that you want to play around with, yeah. And, this isn't rocket science, actually, and lots of other sciences have done this fine. It's just that in life science, we haven't had a tradition of it for so long. So it's not so hard to work this out. The things which get used a lot and have a big impact, you should keep running for a long time. At the start, things should be allowed to fail and fall over, and there needs to be a kind of life cycle of as things that you're like, oh, that worked out well, let's take it up to the next level. That worked out well, let's take it up to the next level. And that includes a kind of death is not quite the right word, but there needs to be a stasis, slow down, and disappearing of these things. So again, I would say this is very much something for when you have a framework where you think about this, and you absolutely acknowledge that there's research in bioinformatics and computational biology that has a faster turnover, but then there's infrastructure provision, which has a very slow turnover. So PDB, as you mentioned, it's been going since 1973. The DNA databases were going since 1982. And so we can think about long-lived storage of this. And for software systems, we can think about virtualization as well for aspects of it. But if we care about it and it has a big impact, we shouldn't worry, I think, about funding it to have an impact over the long term. And as a community, we've just got to get used to that, basically. And ELIXIR is a big step forwards compared to 10 years ago, But I don't think we're anywhere near where our climate change colleagues or the high energy physics community is in thinking about how to have an information infrastructure that matches their science infrastructure and their science direction.
Okay, Ron.
Just a curiosity about this DNA storage business. How much time does it take to retrieve the picture or Shakespeare?
Yeah. So those devices, I don't know where the device is. I hope it's going to come back to me. We'll give you sequence within an hour, maybe 2 hours, slightly depending on the molecular front end to it. And certainly you would be able to do it within a day. Other devices would do it within a day or 2 days.
If you store Shakespeare's sonnet in a book, how much time would you need in order to print it out?
I have to know it by heart. It's Shakespeare.
You have the test tube with all of the fragments.
Yeah.
Now you have to
So the time, the total time. So what you should focus on is the latency, which is the amount of dead time that you, as you know, that you can't do, because we can always improve the total throughput by having more machines. Yeah. So the total throughput is a factor of the amount of money you want to spend on it, but there's always going to be a hard piece of latency, and the hard latency is probably going to be 6 or 7 hours realistically now. And it will get shorter. Those devices will get shorter. But this is not you're not going to use this kind of memory in your laptop. You might use this memory instead of tape drives, or I think you will use this memory instead of tape drives in the future. So storing them for very long-term archival purposes, put them away, keep them in a cold place.
Okay, let's thank Ewan again for a beautiful talk. And